So obviously, eukaryotes are a lot more complicated than prokaryotes, and they have a range of more mechanisms than prokaryotes, which is not surprising. So transcription is essentially you're just going from DNA to, to mRNA that is then translated. But this process is controlled by a lot of gene expression, and we're gonna just go through a couple gene expressions. So we have something called the upstream control elements. The UCE is just 200 base pairs upstream of the start site, and essentially what this does is causing promotion and or inhibiting depending on what it is. And then we have the core promoter region. It's basal transcription. The basal transcription complex will bind to it with the RNA polymerase II binding site. This is located -50 upstream 50 of the core. The core promoter region is -50 or upstream 50 of the start site. We also have the Tata Box. The Tata Box initiates the transcriptional complex and allows assembly of the transcriptional complex. So the Tata Box is -25. It's just a sequence of TATAAA, and this sequence is observed in a lot of eukaryotes. It's either going to promote, or it's going to silence, depending on where it's located at. We have the enhancer sequences, which are located far away from 2,000 base pairs away, or even more. Actually, it could be upstream or downstream. It's not dependent like the upstream control elements or the core promoter regions, which have to be upstream to the start site. This could be the enhancer sequences really can be on either side, upstream or downstream of the start site. So, but the question really comes down to how can enhancers be physically far away compared to other gene regulators and still have some kind of influence on transcription? That really boils down to understanding the loop structure of the DNA, and we know that DNA is coiled around these histines, and because they're so looped up together. For example, we have a loop over here. Let's say that this is a loop, and this is the start site. It might be so far away in terms of nucleotides, but because of this loop, it's still relatively not that far, and the activator proteins, which are essentially bound, the enhancer sequences are bounded by the activator genes. These genes are closer to transcriptional proteins because of this loop structure right here. So gene repressor proteins will inhibit transcription and change in chromatin structure. And eukaryotic transcription is achieved through mass communication. One thing leads to another. So what this really means is that it's a very massive connection of very different things happening at one time due to something. So let's really think about it real quickly. Let's say we had a steroid that came and attached to the surface, or it signals something. Because this steroid is going to signal something to the cell, there are going to be massive changes because of that signal. So it's all kind of integrated and looped up, or like connected to one another. It's all networking in a sense. It's very interesting. But at the same time, this kind of makes us realize that almost all the orders or signals are coming extracellularly. Extracellularly. I can't say that all the time. But yeah, the point behind that is it just kind of shows that these cells are working together and it's responding to the changing environment, and producing what it's being told to make, or responding to the changing environment. So there's a mutation in a cell lipid metabolism causing lipid hydrolytic enzymes to be non-functional. Is this possible with numerous mutations on a single mRNA copy? So what this is saying is that because of this one mutation on a cell or on an mRNA, the lipid hydrolytic enzymes are non-functional. But remember, we talked about this numerously, that eukaryotes don't have polycistronic mRNAs. And that means that more than one gene is not made from the mRNA. Therefore, this is already proved wrong with that sense, and hopefully you see why that's the case. And that's because they're monocistronic. So one gene is created off one mRNA in eukaryotes. So here are some other mechanisms of transcriptional or controlling transcription. So the first is the Rna Translocation. The Rna Translocation really just boils down to understanding that Of course, there's numerous transcriptions happening inside the cell, but they're localized. Some transcripts are sent to certain areas inside the cell, and others are sent to certain areas. So if we think about the neurons, for example, we have specific mRNAs being localized for the soma and the other for the dendroids. So they're all being localized towards their area, and that's one great way of controlling transcription and really just making sure they're headed to the right place. And the other is the mRNA surveillance. The mRNA surveillance is keeping an eye out for the mRNA. It's checking the quality of the mRNA. It's making sure that the mRNA is high quality. If it detects that it's low quality, it's just going to degrade it. It's not going to allow faulty mRNA to just slip out and cause more problems outside. So during transportation of mRNA, they have to be silenced to avoid interacting with. Okay, so here's another very interesting thing. So Rna interference is a process through which miRNA, which is micro mRNA, which is microRNA, and then small interfering RNAs. These will temporarily bind and create a double-stranded RNA. So think about it yourself. You probably know. This will be kind of shocking, not shocking, but it'll be like a very interesting moment. So we know that transcription happens, right? But transcription is a single-stranded mRNA. It's just floating out. So doesn't that mean it's going to interact with other things out there? Because the nucleotides are very, very reactive. And if you remember, we talked about replication of DNA. And when DNA is replicated, we have these single-stranded binding proteins, the SSBP, which will temporarily bind to one of the strands just to make sure it doesn't interact with the environment. So doesn't this mean that the mRNA also has to have temporary nucleotides attached to it? Yes, it does. And that's Rna interference, and this will temporarily silence the gene or the mRNA as it's being transported to the right place. So during transportation of mRNA, they have to be silenced to avoid interacting with wrong molecules. So this can cause translocation to occur in the wrong place as well. So you're working in a biolab, and you got to Stanford and Harvard, you're a smart guy, and you're trying to reduce translational activity for a specific amino acid. What gene regulating method will be most effective, and why? Why? Because if you think about this real fast, all this is saying is that, hey, look, we are trying to silence or temporarily, or as long as you wish, reduce the translation of a certain amino acid. If you think about it, that this can be achieved by RNA interference, because if we have a microRNA or a small interfering RNA attached to the mRNA, it's going to block translation from happening. So you are controlling gene expression right there. So that is the method you'd use over here.